Okay, I'm Oleg Reva from University of Pretoria. What I want to present you today is some results from PacBio project, this very big project, which involves 14 research groups from European countries and from South Africa. It's a EU Horizon funded project. And the idea of this project is to study microbiome of wheat rhizosphere to identify some new bacterial and fungal isolates to improve yield and to replace to some extent harmful chemicals used for fertilization and pesticides. But my personal involvement in this project is lower. I just provide bioinformatics support to the team working in South Africa. So I want to share with you my experience of using long-read sequencing, PacBio sequencing. Okay, so idea of this research, we isolated samples from Rhizosphere at a different winter, cold winter in South Africa, when plants were seeded, and then in October temperature is higher and plants more matured. Then in November, just before harvesting, when temperature is very high and very dry, so drought conditions, and from different fields and from grassland as control. And for every sample, it was in several repeats, of course, and we use these samples for DNA extraction, then followed with metagenomics, targeting 16S RNA and ITS for fungal strains, and whole genome sequencing also was done. Everything was done using PacBio in this case. So what good results from this study? Species richness. Of course, species richness was increased. The ability to cover entire marker region, such as 16S or ITS, by single read, significantly improve identification of species to species level. So we've got a big number of species, fungal and bacterial, and it's approximately 3 times more than when we use Illumina sequencing. This is clear. And this allows us some downstream analysis, like network analysis. So network analysis in the metagenomic involves using a series of samples collected over time or across environmental gradients, like temperature, humidity, for example. And co-occurrence networks are typically constructed based on correlations in taxon abundance, using non-parametric methods such as Kendall-Tau correlation, several other correlation, non-parametric correlation approaches. We use Kendall correlation. Because many species we have, here you can see several examples of networks. We've got much more networks. But what is the idea? Every node is one species, bacterial species or fungal species. They interconnected by links. Red links, it's negative correlation. Green links, positive correlation, and some strains are highlighted. Red is potentially phytopathogenic organisms, green potentially beneficial organisms, and you can see several nodes represented by bacterial and fungal species interconnected with many other species. They are central nodes which may be used as markers of alternative community members, either associated with pathogens or with beneficial bacteria. What next? Genome assembly. Another area where long-read sequences may help. I agree with previous presenter completely. And we use our pipeline based on available tools. Start with this pipeline. Basically, workflow is good for short read, for long read, but different programs are used. We start with quality control by Tribomatic for short read, for long QC for long read, and then pipeline combined together de novo sequencing and resequencing by mapping against identified reference genomes, and program RAHTAG make scaffolds and put together this result of these two approaches. As a result, we identified a quite big number of contigs longer than 10,000 base pairs, and some of them we identify taxonomic provenance using first annotated them and then use diamond to identify to which bacterial fungal species they belong to. After that, we combine several not too bad marks from these contigs. But because we have contigs and they were annotated, we can now identify, we created our database of genes which are involved in important processes for soil fertilization, like nitrate fixation, ammonification, phosphate mobilization, polysaccharide degradation, and just blasted all these genes against our local database, identified some key species which are enriched with good genes for us. We can guide further isolation of the species from soil and use them in field trials. Okay, I was rather fast with these slides because this is generally classical metagenomics, some variants related to projects, but this is all understandable. Now we come to one point that I love the most. This is epigenetics, bacterial epigenetics, and it's a very new topic of research, so new that in this big conference like ours, I didn't find any single talk or any single presentation on bacterial epigenetics. Don't mention use of epigenetics in metagenomics. It is a pity because it's very useful tool. One publication we published several months ago is about methylation. Basically, when we are talking about epigenetics, very often we mean methylation, metylomics. And in this case, indeed, all bacteria, many bacteria have methylation profiles. And in this example, we use 4 closely related Staphylococcus aureus, which are have very conserved genomes, but they have different methylation motifs at which methylation take place. So even we can use this for surveillance of organisms released to field, for example. Is it the same strain which we released, or maybe some closely related organism? So we can distinguish them by using epigenetics. But metylomics is only part of epigenetics. Another important part of that nucleotides can be modified in other ways. For example, here you can see guanine oxidation, which happens very often for guanine to 8-oxoguanine. It's a very dangerous process because 8-oxoguanine is really create mutations for next generations. So bacteria have to keep under control the frequency of oxidized guanine. The enzymes, there are specific enzymes which control this process, but when bacteria is under stress, it's not so effective with control of oxoguanine. Like, look here, this is bars on the right part, blue bars. This is the frequency of oxidized guanine in one bacterium, Florobacterium, which was always abundant in soil samples. At the beginning, cold weather, rainy weather, some level of oxidized guanine. Then number 3, it's October, better conditions is drop. But now look at number 4. This is drought conditions. It's extremely increased number of oxidized guanine. Bacteria is present, is abundant, but is stressed. Let's check, do the same analysis with different species. And you can see that what is good for one species is not good for another species. Several species didn't tolerate flooding, cold weather, but then regain. But other organisms, flooding was okay, a lot of wet is okay, but drought conditions is not good for them. And why this is important? Because if we want to isolate, identify some bacteria to use as, we have to introduce them in optimal conditions. Plus to do this method provides us with fantastic opportunity because now we can bacteria which are under stress, not necessarily we can see abundant shift. They still can be abundant. They can be even dead, but DNA is still in samples. Here we can distinguish between bacteria who thrive and bacteria who struggle with survival. And we can use in many approaches. For example, in gut microbiome, if we use antibiotic, we can check to which bacteria it's no problem, which is not good. Okay, so there is another program which we published a few weeks ago, and where we explain which way can be used to visualize distribution of modified nucleotides and do some statistical analysis. And okay, some conclusion. Long-read sequence reduce misclassification, improve assembly quality, frequency of oxidized guanine in sequence genome indicates stress conditions, and new software tools were provided for you and published. I want to acknowledge contribution of my colleagues, my students, and all participants of Tribiome project. Thank you very much.
Thank you. So we have time for one quick question. And I'm also just going to ask who the next speaker, if you can just come on over if you have a quick question.
Okay.
So if there's a question,
no questions. Thank you
so much. Yes, we don't have very big time.